Welcome Jan to Nepal on behalf of the Center for Molecular Dynamics Nepal, CMDN, and KIPS 98.3 FM.
Thank you, Devesh. I'm very excited to be here speaking with you. I'd also like to thank CMDN for inviting me down here to Nepal.
We are delighted as well to have you here, both in our studio here as well as in our organization, sharing your expertise in wildlife genetics in particular and wildlife conservation in general. So, could you give us a little bit background about yourself?
Yeah, sure. So I'm at Texas A&M University right now. I have been studying mostly evolution of mammals. And then over the last 4 or 5 years, I have focused on using genetic tools to look at wildlife, and particularly to look at the status and distribution of populations, and then how we can use genetic tools to better understand landscape genomics and movement between different areas, and to provide information that is important for conservation of species. And so I'm very excited to be here to share ideas and to work on ways we can work together to understand wildlife in Nepal.
Good. Now, I believe you have done some work, some significant work on snow leopard. And could you give us an idea in terms of how you developed the genetic tools to kind of do the study, and how did that help you in comprehending different aspects of conservation?
Mm-hmm. So the snow leopards are a very unique species, and they're one of the cats that there's very little known about. So of the big cat species, which include the tiger, the African lion, the leopard, the jaguar, the snow leopard has the least amount of information on it. And it's distributed throughout Central Asia, the mountainous regions. And because it's found in such rugged and remote areas, there's very little information that we have on the species. And it's therefore of high conservation priority to get information on population status and abundance, and also the ecology of the snow leopards. And so traditionally, what people have done is to use camera trapping, where they go out into the field, they put cameras that have a remote sensor on them, and then when an animal walks by, it takes a picture. And so they've used this technique to estimate abundance and density of animals such as tigers in India. Another species that this technique has been used for is ocelots in South America. But the problem is with snow leopards, because the areas are so remote and the mountains are so rugged, it's very difficult to conduct these kinds of surveys. And so the other way that people look at species and to look at species ecology and population status and abundance is to, of course, do direct observational studies. So people have done this, researchers have done this in Africa with, for example, African lions. And then the third approach is to use telemetry. Mm-hmm. And so researchers catch animals with some kind of either a leg hole snare that's modified to be safe for the animal, so it's not injured during the trapping, or live box traps, and they sedate the animal, put a radio collar on it, and then release it into the wild, and then it provides a signal that can be used to track the animals. Both these approaches are very difficult to use for snow leopards. So of course they're very elusive animals. They're very rarely seen. Some of my colleagues that have worked with snow leopards for 15, 20 years, they've only observed them several times in their life, once or twice, even though they spend a lot of time in the field. So observations don't work too well, direct observational studies. The other study that I mentioned, the radio telemetry, is also very difficult because it's very hard to actually trap these animals. And in addition, you have to spend a lot of time in the field, and it's very difficult in the remote, rugged areas of Central Asia. And so we tried to develop or to apply some of the genetic techniques that have been developed in the last 10 or 15 years for monitoring wildlife towards snow leopards, to see if we can use this method to understand snow leopard distribution, abundance, and ecology. And so what this method does is it uses very sensitive DNA assays to sample populations of snow leopards by collecting samples non-invasively. And so basically we have these methods where we can go out in the field and we don't have to observe the animals directly, but we can just collect a non-invasive sample. And so primarily with snow leopards, what we've been doing is collecting their feces, or what's referred to as scat. And so we can go out there, survey wildlife trails over a fairly short period, collect the feces, bring it back to the lab, and then extract the DNA, and then do techniques similar to what's used in forensics for identifying, for example, samples of blood at a crime scene, and to basically use that DNA fingerprinting technology to identify individuals. And so we started these surveys initially in India in 2005 and then China in 2006. And so we basically tried to optimize these methods to see how they can be used effectively for monitoring snow leopards.
Right. And when you like, we are basically exploiting the unique nature of DNA for each species, and then also doing DNA fingerprinting on individual snow leopard, for example, to kind of differentiate between one snow leopard versus the other, right?
Yeah, that's right. So there's a couple different methods that can be used. You said first is to look at the difference between species, and so this is actually a critical part because it's very difficult to identify the feces in the field. Previously, biologists thought that it's fairly easy to visually identify snow leopard scats, but we found it's sometimes misidentified with even red fox, which would, from a general sense, they would seem that they shouldn't really look so similar, but they do. And so we actually sequence a portion of genes that are in the mitochondria of cells, and we use that as a sort of a marker to determine which species left that scat. Then what we've done recently in the lab too is developed a assay that only works on snow leopards. So these are what's called PCR primers, are developed to only amplify DNA for a particular gene or fragment of a gene in the snow leopards. These can be used in assay to do species identification.
Right. So you're looking for the special segments within the snow leopard genome to kind of identify them, and then kind of doing subsequent tests to kind of see if we can do a DNA fingerprinting on each snow leopard. Yeah,
that's right. And there was actually recent developments in the last 3 or 4 years. They did whole genome sequencing of the domestic cat and developed these genomic maps where basically they tell you what part of, or I should say, what genes are found on what chromosomes, and sequence those. And what we found is that the genome structure in snow leopards is very similar to domestic cat. So we used the domestic cat whole genome sequence and some of the genetic linkage and radiation hybrid maps to develop the markers for snow leopards that can be used for the individual identification.
Right. And then going back to more of a general question, how do you like Nepal? You've been in Mongolia and then in India as well as some other Asian countries. How do you find Nepal?
I really liked it here. I spent the last week was excellent. There's a couple of reasons why I've been really impressed with it, and I've really enjoyed being here for the last week. The first is of course culturally. There's I haven't had too much time to see a lot of areas that I would like to see, but some places I've been, this incredible, the Hindu and the Buddhist temples and sites, and I'm very interested in art and and craftsmanship and stuff like that. So it's been a real pleasure to learn about that sort of thing, and also some of the philosophical ideas that are here. And then of course the other important aspect why I really like Nepal is that I've really enjoyed the people that I've worked with, particularly your CMDN. And I found that actually the lab I was very impressed with. The technicians are highly skilled and qualified, and actually, compared to some of the other labs that I have visited in China, this is really the best so far that I've seen out here.
Well, thank you very much for that. Now.